Good afternoon, or good morning, depending on your location. Welcome to the following webinar entitled Stem Cell Culture, Optimizing Expansion and Differentiation. My name is Matthew Riolo, and I'm a technical writer and product manager for GenScript. I have a PhD in pathobiology, and later I worked in drug development focused on small molecule inhibitors. As a graduate student, much of my work focused on isolating hematopoietic stem cells and differentiating them into dendritic cells and macrophages for downstream analysis of innate immune pathway activation. I'd like to thank you for joining me, and I encourage you, if you have any questions, to type them into the question box on the bottom of your screen, and I will address them following this presentation. Any questions I do not get to, I will post on GenScript's website in the days following this webinar. So I'd like to give you a quick background on GenScript. We are one of the world's largest biological service companies and a leading provider of research products for laboratories in areas of immunology, molecular biology, and translational drug development. As one of GenScript's product managers, I oversee our portfolio of recombinant proteins and antibodies, which we are constantly updating. So please, don't hesitate to follow up with me if you have any questions on the products after the webinar is finished. So this next slide gives a nice overview on the topics I'll be covering today. First, I'll start out with a very broad overview of collecting and expanding hematopoietic stem cells, which I'll refer to as HSCs throughout the rest of this talk. I will then give some specific tips and examples on how to optimize differentiation of HSCs into different types of dendritic cells and macrophages. Following this, I will switch gears and talk about embryonic stem cells, specifically best practices to expand undifferentiated embryonic stem cells, as well as how to promote orderly differentiation into embryoid bodies, and what types of culture methods will promote differentiation to neurons, pancreatic cells, and hepatocytes. This is a fairly new field of research that is constantly evolving. I will cite specific publications that will demonstrate how timing, combined with combinations of growth factors and cytokines, will lead to differentiation of your desired cell type. So before I begin the technical part of my talk, I'd like to start off by highlighting some advantages of stem cell research. Now, from a medical point of view, expanding HSCs for autologous reinfusion to treat leukemia, certain immune disease, or solid tumors could have great promise once optimized in the future. From a more basic sense, neuroscience research has shown recent reports of scientists generating dopamine and motor neurons showing some initial success when reimplanting them into mice, which leads to improvements in cognition. This could open the door to new strategies in treating Alzheimer's disease, Parkinson's disease, and other neurodegenerative diseases. Throughout this talk, I will discuss strategies that help optimize differentiation of HSCs into immune cells, as well as differentiation of embryonic cells into neurons. So I'll begin the first part of my talk discussing strategies to optimize expansion and differentiation of HSCs. Now, HSCs are incredibly valuable for stem cell and immunology research because they are the starting point of all immune cells, and they are blood cells that possess both self-renewal and multipotencies. In essence, what this means is they can become any immune cell under the right circumstances. Part of our goal as researchers is to determine what type of environment ex vivo would best represent the environment in vivo and would allow these HSCs to thrive, as well as differentiate them into our desired cell type. Now, while there are various sources for obtaining HSCs, there are two main sources that stand out: cord blood and bone marrow. Now, cord blood is obtained from umbilical cord and has several advantages to bone marrow. First, it's highly enriched with the HSCs and tends to have a higher amount than that found in bone marrow. Also, there's a convenience factor. Cord blood is readily available from several biotech providers and can be purchased when needed. Third, it's the preferred source when doing human studies. There's also a therapeutic advantage. Now, many individuals are having their cord blood frozen and banked in case it is needed for treatment later in one's life. Doing this provides two benefits to the individual. One, because there is no donor, there is significantly low risk for any type of graft versus host disease. And two, there is almost no risk for transmitting infectious disease. Now, the main source of bone marrow HSCs is from the femur of mice. The advantages of using a mouse system is the ability to use knockout mice to investigate the requirement of certain genes for immune responses. Also, using a mouse model allows for study of transplant system and testing the effects of ex vivo manipulation on xenograft tumor models. Using mice are a good starting point for drug discovery and testing how regeneration of cells may affect the host system when they are retransplanted. So this slide illustrates the first stage of isolating HSCs. Now, starting on the far right is obtaining HSCs from cord blood. Now, if you're using cord blood, you're most likely obtaining it from a licensed vendor, so basically the first step is just to thaw the blood when ready and transfer it to a tube. Now, on the far left is a femur bone from mice. The two most important steps for optimizing bone marrow is to clean cut on each joint. As you can see on the dotted lines, you'll want to use a broad scissors and cut smoothly and firmly along the top joints. A clean cut will prevent bits of bone from entering your sample, and it will also provide optimal access to the marrow, which will make it much easier to flush out. Now make sure you flush out both ends twice with media. Now once the bone marrow or cord blood is collected in a tube, you'll want to isolate HSCs by separating out CD34 positive cells. Now the reason we want to isolate CD34 positive cells is because CD34 is a cell surface marker for undifferentiated cells characteristic of HSCs. Now by only culturing CD34 positive cells, you will optimize the amount of HSCs you expand per culture dish, thereby maximizing your final yield of differentiated immune cells. Now isolating CD34 positive cells starts with running cord blood through what is known as a Ficol density gradient. By adding anticoagulants to your blood sample and then adding the blood to a 50 ml tube containing the Ficol, you could separate out the contents into distinguishable layers through a series of centrifugations. Now in the first centrifugation, peripheral blood monocytes will position between the plasma and Ficol as depicted here in the diagram. Carefully aspirate out the top layer of plasma and then transfer these PBCs to a fresh tube. Then centrifuge this tube and remove the supernatant. To optimize the number of CD34 positive cells, the pellet is resuspended, centrifuged again, and the remaining supernatant should be aspirated. The pellet should then be resuspended and added to the tube. This resuspension should be passed through a nylon mesh to get rid of clumps, and then magnetic beads should be added. The magnetic beads will have antibodies for CD34 and attach to the cells, forming aggregates. To isolate these bead and antibody aggregates, simply place a magnet next to the tube. Now when collecting and isolating CD34 positive cells, several protocols will use IMD media with 10% FBS. However, many of these protocols switch to a serum-free system when expanding and differentiating HSCs. The reason for this is that the composition of FBS may not always be exactly the same from batch to batch. Even small differences in growth factor composition can cause variability in experimental results. Also, FBS may contain growth factors that impede stem cell growth, such as TGF-beta. Finally, there is a risk of xenogenic contamination if culturing human stem cells in the presence of FBS. Now, while it is a good idea to refrain from using FBS in HSC culture, there are 3 cytokines that are critical for optimal expansion and differentiation. Now, these 3 cytokines are listed, and they are stem cell factor, thrombopoietin, referred to as TPO, and FLT3 ligand. Now, SCF and FLT3 ligand bind to receptors on CD34 positive cells that promote HSC expansion while inhibiting apoptosis. Now, TPO binds the receptor involved in platelet development, and studies have shown that it synergizes with SCF in promoting healthy HSC growth. Additionally, studies demonstrate that combinations of SCF, TPO, and FLT3 ligand is critical to expanding and maintaining dendritic cell precursors. Therefore, using this combination of cytokines to expand your HSCs will help improve and optimize your dendritic cell yield even prior to beginning differentiation. Now in the next few slides, I'm going to focus on strategies for optimizing differentiation and yield of the following immune cells. Now classic dendritic cells are referred to as myeloid dendritic cells, and they are critical in the immune response as antigen-presenting cells that secrete pro-inflammatory cytokines in response to infection. Now the less abundant plasmacytoid dendritic cells secrete large amounts of interferon alpha in the immune response. Lastly, macrophages, which phagocytize foreign agents, can secrete either pro- or anti-inflammatory cytokines based on their polarization due to microenvironmental stimuli in combination with their epigenetic composition, which I will touch upon in a future slide. So we'll first start with a basic culture scheme for optimizing your dendritic cell yield from HSCs. Now, once you've isolated your CD34 positive cells and placed them in serum-free media, you'll want to add your cytokine expansion bundle consisting of SCF, TPO, and FLT3 ligand. Now these cells should be cultured for 7 days under these conditions, replacing the media with a fresh cytokine bundle every 3 days. Now on the 7th day, the cells should be split and passed to new plates with media containing SCF, FLT3 ligand, and now the addition of GM-CSF and IL-4, interleukin-4, which will promote myeloid dendritic cell differentiation. Now in the last 48 hours, these dendritic cells should be stimulated with TNF-alpha to ensure maturation, at which point they can be collected for experiments. Now, this protocol is geared toward human hematopoietic stem cell culture from umbilical cord blood. Now, while it can be applied to mouse HSCs, many protocols with mouse HSCs do not require the addition of SCF, TPO, or FLT3 ligand, therefore skipping the first 7 days of expansion and starting at the point of adding GM-CSF and IL-4. Now, in addition to SCF, TPO, and FLT3 ligand, some protocols suggest adding either interleukin-6 or interleukin-3 into your HSC expansion bundle, which will enhance your final dendritic cell yield. Now, to identify how IL-3 or IL-6 influences dendritic cell differentiation, a group of scientists tested using serum-free media the cytokine bundle of SCF, TPO, FLT3 ligand, in the presence of either IL-3 or IL-6 to see differences in differentiation. Now, what the scientists observed was that the addition of each cytokine had a preferential influence on what type of dendritic cell progenitor was generated prior to dendritic cell maturation. Now, the addition of IL-6 generated a preference for dendritic cell precursors exhibiting a phenotype toward dermal DCs as opposed to precursors of Langerhans cells. Now, on the other hand, the addition of IL-3 showed a preference toward a precursor phenotype of Langerhans cells, inhibiting the generation of dermal DC precursors. However, following the addition of GM-CSF, interleukin-4, and TNF-alpha, both IL-3 and IL-6 generated precursors differentiated into mature myeloid dendritic cells. Additionally, the scientists observed that IL-3 in the presence of SCF, FLT3 ligand, and TNF-alpha led to the generation of plasmacytoid dendritic cells, which was confirmed by their production of high levels of interferon. Now, the more standardized way to differentiate plasmacytoid dendritic cells, known as PDCs, from expanded HSCs is simply culturing them in IMDM or RPMI media supplemented with FLT3 ligand for 7 to 9 days, replacing the media with fresh cytokines every 3 days. Now, it's critical that you do not add GM-CSF to your PDC culture, because this will immediately provoke a preferential differentiation into classic myeloid dendritic cells. However, some protocols suggest that you do add G-CSF to HSCs that will help promote PDC differentiation in the presence or absence of FLT3 ligand. Now, in my personal experience, I always just used FLT3 ligand and got a decent yield of PDCs from bone marrow. However, I did find that my myeloid DC yield was always higher than my PDC yield of the same batch of cells. One other important note regarding PDCs, that is, if you're investigating changes in interferon production for your experiments, you'll want to make sure you stimulate with CpG2216, not CpG1668. Now, unlike splenic PDCs, FLT3-derived bone marrow PDCs will not produce interferon in response to CpG DNA 1688. However, FLT3-derived PDCs will produce interferon in response to CpG DNA 2216. Now, when differentiating HSCs into macrophages, the key growth factor to use is MCSF, and there are two main culture strategies. The first seen here at the top is using media from cultured L929 cells. Now, L929 cells are a mouse fibroblast cell line that can be obtained from any major cell line vendor, and when cultured, secrete significant amounts of MCSF. Therefore, one strategy is to culture several plates of L929 cells for 7 days and collect the MCSF conditioned media. The media should then be filtered and then either stored at -80 degrees or used immediately for the culture of HCS cells to differentiate them into macrophages. The other strategy to differentiate HSCs is by adding recombinant MCSF directly into the cell culture media, such as rPMI or MEM. Now, a typical amount of MCSF for macrophage differentiation is around 50 nanograms per mil. However, this can differ based on the bioactivity of your cytokine. And these cells should be cultured 7 days. Now, regardless of which strategy you use, it is important to make sure that you change and add fresh media at the end of day 3 or the beginning of day 4 to ensure healthy macrophage differentiation. Now, macrophages can be divided into two separate pools, M1 and M2. Now, M1 macrophages are classically activated and execute an acute pro-inflammatory Th1 response targeting bacteria and other pathogens. M2 macrophages are alternatively activated. They are anti-inflammatory and they aid in wound repair. They can also help regulate the immune response by counteracting M1 macrophages and participating in the Th2 response. Now, M2 macrophages are further subcategorized into 3 populations, M2A, M2B, and M2C, which is defined by their surface molecule expression and genotype. Now, what is important for the culture purposes of macrophages is that different strategies should be implemented depending on which type of macrophage one wants to study. Now, there has been some debate as to what combination will produce a specific population of macrophages. Classically, monocytes would be differentiated by MCSF for 5 to 7 days and then polarized to either an M1 or M2 phenotype with the addition of cytokines in the last 24 to 72 hours. Now, for M1 polarization, interferon gamma would be used, while M2A polarization should include the addition of interleukin 4 and interleukin 13. Now, for MB polarization, treat with LPS or add interleukin 1 beta. M2C polarization requires the addition of IL-10. Now, most protocols use MCSF to differentiate HSCs into macrophages regardless of the polarization. However, there has been debate suggesting that culturing macrophages in GM-CSF instead of MCSF results in M1-like macrophages expressing high levels of IL-12 and low IL-10, while using MCSF will induce M2-like macrophages that have low levels of IL-12 and high levels of IL-10. Now, to help resolve this debate, a group of researchers tested the effect of MCSF versus GM-CSF on macrophage differentiation and polarization. So the scientists used 4 different culture conditions to test the epigenetic background and activation response of the resulting differentiated macrophages. Now, the 4 culture conditions were MCSF for 7 days, followed by either interferon gamma or interleukin-4 and interleukin-13, or GM-CSF for 7 days, followed by interferon gamma or interleukin-4 plus interleukin-13. Now what the scientists found was an initial distinct phenotypic difference between the cells cultured with MCSF versus GM-CSF. Now with MCSF, the phenotype was CD14 high, CD163 high, and CD206 intermediate, while with GM-CSF, macrophages exhibit a phenotype of CD14 low, CD163 low, and CD206 high with an elevated interleukin-12 expression. However, further polarization into M1 or M2 macrophages with the treatment of interferon gamma or IL-4 plus IL-13 generated similar genotype markers, suggesting that this polarization is really regulated at the transcriptional level in response to either interferon gamma or IL-4 plus IL-13, as opposed to whether the macrophages are differentiated with MCSF or GM-CSF. So that concludes my discussion on the expansion and differentiation of HSC cells. Now I'd like to move on to embryonic stem cell differentiation, and I'll start with an interesting timeline. Something here that is interesting is that the first stable mouse embryonic cell line was discovered 30 years ago, and now there's been a 20-year gap for the first human embryonic stem cell line. Now, about 8 years ago, there was an identification of key growth factors that are critical for the undifferentiated expansion of human embryonic stem cells. What I will touch upon in later slides are 2 case studies focusing on hepatocyte differentiation and pancreatic cell differentiation. So for many labs, isolating embryonic stem cells will entail purchasing them from a vendor. Now, vials should be received frozen on ice packs and stored at -80 degrees until ready for culture. Now, before thawing, you should already have a 15 ml tube with 8 to 12 milliliters of warm media ready, as well as culture plates with media and a cell culture hood. Now, thaw the vial of cells at 37 degrees and then transfer to the media in the 15 ml tube. You'll want a centrifuge between 5 to 10 minutes at between 1,000 to 1,200 RPM. You want to remove the supernatant and then resuspend with 10 mL of media and transfer to a fresh culture dish. Now, as this will grow healthy embryonic cells, they'll form colonies with a distinct morphology. You'll be able to distinguish the healthy embryonic cells because they'll have a high nucleus to cytoplasm ratio, and they'll be tightly packed within each colony. Now, these distinct colonies should be spread out throughout your culture plate, and they should not overlap or touch each other. Now, if the embryonic cells are cultured on a feeder cell line, you should see a distinct edge between where the embryonic cell colony ends and when their feeder cell begins. Now, on average, for a 10 ml plate, you should have somewhere between 60 to 100 colonies. Now, once the embryonic cells are plated, it is important to change the media frequently. I can't stress this enough. A safe bet is to change media every day. Also, you don't want to let your embryonic cells become too confluent or remain in the same culture dish for too long. So split your embryonic cells every 2 to 4 days. It is critical that embryonic cells are healthy when splitting. Now, if you notice at the bottom of this slide, if the media starts to change color to an orange or yellow, then differentiation is starting to occur, and these cells should not be split. However, if you split your embryonic cells regularly and use the right combination of growth factors and cytokines, you can expand your undifferentiated embryonic stem cells as long as you like. I strongly encourage to freeze back large pools of embryonic stem cells within the beginning passages. Now, while theoretically they should expand undifferentiated with the right cytokine and growth factors for several passages, once you get over 20 passages, this can start to raise your risk of karyotype mutations. So what culture reagents should you use to optimize undifferentiated embryonic cell expansion? Well, that depends on several factors that we'll go through. First, are you using mouse or human embryonic stem cells? Now, if you're using mouse, you want to supplement your media with leukemia inhibitory factor known as LIF. Now, what LIF does is it binds to the LIF receptor, activating JAK-STAT and MAP kinase pathways. Now, most embryonic cells are obtained from the inner cell mass, which express high levels of LIF receptor. Now, when they are removed and used for culture, we need to provide that LIF that they would normally receive for their in vivo environment, and this will prevent differentiation. In addition to LIF, it is also recommended to culture most embryonic cell lines on a MEF feeder cell line. Now, MEF feeder cells will secrete several factors that will contribute to the prevention of differentiation. Now, for human stem cells, you may want to consider what type of study the cells are going to be used for once they're differentiated. If you're using cells for preclinical study to study the effects of a drug or to optimize differentiation of cells that could lead to new strategies for reimplantation, then you may want to consider not using a MEF feeder cell line. Instead, there are a few human feeder cell lines that have shown to be effective in preventing differentiation, or you can try go feeder free. If the cells are for more observational studies, like mechanistic signaling, or something pertaining to basic biochemistry or molecular biology, then you may not be so concerned with using a feeder cell line. Next, you'll want to decide on the best media. RPMI, DMEM, or vendor-specific media all have been shown to be effective in expanding undifferentiated embryonic cells. A more pressing question is whether you want to use FBS or not. Now, many studies are pushing toward a serum-free media to avoid possible xenogenic contamination and variability in results. If you find that without FBS, your human embryonic stem cell growth is reduced, you could try supplementing the media with BSA, or if you have to use FBS, try testing the lowest amount you can use and then use that for your future expansion. Now, regardless of whether you use a feeder layer or FBS on human embryonic stem cells, you will need to culture them with fibroblast growth factor basic. In addition to fibroblast growth factor basic, you will need to inhibit the born morphogenetic pathway which promotes differentiation into human embryonic stem cells. Now, to do this, you want to supplement your media with the protein noggin. Now, some studies suggest that you can use a low dose of noggin with FGF basic, while other studies recommend using a high dose. However, this may be dependent on what type of human embryonic cells one is using, so it's a good idea to check with the vendor first or try different strategies before optimizing your protocol. So as I mentioned, depending on your downstream differentiation and future experiments, you may or may not want to use a MEF feeder cell line when expanding undifferentiated embryonic stem cells. So what is a MEF feeder cell line? MEF feeders are mitotically inactivated by gamma radiation. In culture, they secrete several growth factors, cytokines, and extracellular matrices that support the expansion of undifferentiated embryonic cells. However, there are some negative aspects of using a MEF feeder cell line. First, there is the potential for xenogenic contamination, especially when using human embryonic cells. Also, there may be some variability in what factors are secreted from MEF feeders from experiment to experiment. Now, as a researcher, you have more control in standardizing exactly what factors supplement your media when you use individual cytokines and growth factors, as opposed to using a biological source such as a MEF feeder cell line. Now, our goal is to reproduce the environment and culture that is optimal for stem cell expansion. Now, this could be difficult to pinpoint exactly what type of cytokines and growth factors are excreted from MEF feeder cell lines that provide this environment of undifferentiation. However, studies do suggest that fibroblast growth factor basic and noggin help promote undifferentiated embryonic expansion, as well as there are a few human feeder cell lines that can help eliminate the risk of xenogenic contamination. So this is just a quick review of the differences between undifferentiated expansion of human versus mouse embryonic stem cells. As we said, with human, you're going to want to culture your cells with FGF basic, and you'll want to use noggin to inhibit BMP pathways. Now with mouse, to expand undifferentiated embryonic cell lines, you're going to want to culture with LIF, and some studies have suggested that actually including BMP protein will help expand undifferentiated mouse embryonic cells. So moving on from undifferentiated expansion, I want to talk about approaches to differentiating embryonic stem cells into a desired cell type. Now for neurons, there are two approaches. Approach one is to imitate the neuroectoderm environment and then provide cell-cell contact influencing embryoid body formation. Approach two is to provide no cell contact, and then as a default mechanism, it will induce neuron differentiation. Now most studies incorporate both strategies, and a very important part of differentiating stem cells into neurons or other types of cells is to induce embryoid body formation. So what are embryoid bodies? Well, embryoid body formation is the initial step of embryonic stem cell differentiation. Now to do this, first, any factor that is preventing differentiation, such as FGF basic or LIF factor, should be removed. Now, once these factors are removed, embryonic stem cells will begin to differentiate and form spherical aggregates known as embryoid bodies, which consist of future endoderm, mesoderm, and ectoderm tissue. Now there are various methods to regulate the formation of embryoid bodies. The first I will discuss is the transfer of embryonic cells into a suspension liquid in a bacterial grade plate. Now while this does form embryoid bodies, it is not ideal. The reason for this is the embryoid bodies will be heterogeneous in size. One way to remedy this is to place the bacterial suspension disc on a rotating device. However, a better and more common method is known as the hanging drop method. Now basically, this strategy is to take droplets from your original culture of differentiating embryonic cells and place them on the top of a lid in a culture dish. Now the drops should be uniform in size, and this will ensure that each embryoid body will have the same amount of embryonic stem cells. Once the droplets are placed on the lid should be inverted over a petri dish, and what will happen is the embryonic cells will start to form these droplets. Now these droplets will cause aggregation, which will eventually form into embryoid bodies. To ensure this, you should put the lid on top of a petri dish and incubate it for 2 days. Now in the petri dish, you should have PBS. This will prevent the embryoid bodies from drying out. Following this, you'll get a homogeneous sized embryoid bodies that can then be transferred to the culture dish suspension and cultured for an additional 3 to 5 days. Now there is an alternative method, is using a methylcellulose culture, which is another good way to form embryoid bodies, because what will happen is one cell will end up forming aggregates that will generate a homogeneous embryoid body from that one cell. Now this provides highly reproducible embryoid bodies. Two other methods are simply using a round bottom tube or a 96 well round bottom plate. And what will happen is when you transfer the embryonic stem cells to the round bottom tube or 96 well plate, they'll form aggregates in the bottom that will eventually become embryoid bodies. So here are a few common culture systems that can be used to differentiate embryonic stem cells into different types of neurons. Now on the top left, the top left highlights using stromal cells as a meter of a feeder cell line, which promotes human embryonic stem cells to differentiate into neural rosettes, which are of a neural epithelial lineage that can be further differentiated into motor neuron progenitor cells by culturing with sonic hedgehog and retinoic acid. Now the top right box highlights the use of Matrigel as an extracellular culture system that supports the differentiation of human embryonic stem cells into neural precursor cells by providing an environment that promotes neurite growth and survival. Now a third option for your culture system is differentiating embryonic cells into neurons using conditioned media. Now this method is like the previously described method macrophages using the L929 conditioned media. Or you can culture, what you'll have to do is culture stromal and fetus cell lines separately, collect the media, filter it, and then use it to differentiate your embryonic stem cells. This will reduce the risk of direct mouse to human cell contact. So this next slide shows some culture methods to go from human embryonic stem cells into different types of neural lineages and differentiate into different categories of neurons. First, stem cells should be allowed to form the embryoid bodies we talked about earlier by culturing in the absence of factors that inhibit differentiation, such as FGF-basic or LIF. Well, FGF-basic for human. Now, the addition of retinoic acid for a few days of culture will induce the human embryoid bodies to form neural rosettes as described in the previous slide. Now, these neural rosettes can be further differentiated into either motor neurons or dopaminergic neurons based on the combination of growth factors added. Now, as I just described, adding sonic hedgehog to this culture in the presence of retinoic acid will promote motor neuron progenitor cell differentiation. From there, adding brain-derived neurotrophic factor in combination with glial-derived neurotrophic factor and insulin-like growth factor 1 for an extended period of time will differentiate these motor neuron progenitors into mature motor neurons. Now, to differentiate the neural rosette cells into midbrain dopaminergic neurons, they should be cultured with the addition of basic fibroblast growth factor, then fibroblast growth factor 8 with sonic hedgehog. Now, the second method is to differentiate embryoid bodies into oligodendrites, which are non-neural cells of white matter that support the central nervous system. Now, one method that scientists have shown to be effective is treating embryoid bodies in the absence of FGF-basic while in the presence of insulin, transferrin, selenium chloride, and fibronectin to promote differentiation into neural progenitors, which were then cultured in basic fibroblast growth factor, promoting differentiation into neural precursors. The neural precursors are then transferred to Matrigel and treated with epidermal growth factor and platelet-derived growth factor, differentiating these progenitors into early oligodendritic precursor cells, which was verified by specific cell marker expression. Now, at this point, the growth factors are removed, and the cells are treated with thyroid hormone, which forms mature oligodendrocytes. Finally, the third culture method shows embryoid bodies can be differentiated into retinal progenitor cells through a combinatorial treatment of noggin, DKK1, and insulin-like growth factor. Upon formation of retinal progenitors, they are transferred to Matrigel-coated plates in the presence of basic growth factor. After several weeks, the cells demonstrate expression of transcription factors specific to retinal neuron differentiation. So now I'd like to focus on a recent case study where scientists differentiated human embryonic stem cells into pancreatic endoderm cells through 4 distinct stages. Now, in the first stage, scientists used serum-free chemically defined media with a combination of growth factors they referred to as FAB for FGF-basic, activin A, and bone morphogenetic protein 4 on human embryonic stem cells that were grown on a MEF Feeder cells in a 6-well plate. Now, by day 4, these cells demonstrate definitive endoderm formation. Now, following this, the scientists transferred the cells to suspension culture plate to induce embryoid body formation in ITS media supplemented with basic fibroblast growth factor. Now, after 14 days, these embryoid bodies demonstrated gene expression of PDX and FOXA2, which is indicative of early proliferative foregut endoderm and pancreatic lineage. Now, the embryoid bodies were then isolated and cultured in matrigel supplemented with serum-free media containing insulin, transferrin, selenium, FGF7, nicotinamide, and islet neogenesis-associated peptide, as well as exendin-4, which is a long-acting GLP-1 agonist. Now, between 21 to 28 days, the embryoid bodies differentiated into what scientists refer to as PDX1 positive clusters, which express high levels of the PDX1 gene and proteins indicative of islet cells. In stage 4, the media is changed to RPMI containing B27 and nicotinamide for 7 to 14 days, resulting in a 10-fold increase in insulin-producing cells. So finally, I'd like to just give a case study that focuses on the differentiation of human embryonic stem cells into hepatocytes. Now first, the human embryonic stem cells were differentiated into definitive endoderm and low serum RPI media on a MEF feeder layer. The cells were then cultured in the presence of activin A, which is known to bind to nodal receptors, thereby inducing endoderm differentiation. Now, following 5 days of culture with activin A, the stem cells lost expression of the pluripotent OCT4 while demonstrating expression of definitive endoderm transcription factors SOX17 and FOXA2. The definitive endoderm was then placed on a collagen matrix-coated dish in RPMI media, supplemented with knockout serum and 2 growth factors, FGF4 and HGF. The cells grew on the collagen matrix as a confluent monolayer, and after 3 days, the media was changed to minimal media, supplemented with BSA with the same concentration of FGF4 and HGF. Now, after an additional 3 days, the media was changed to complete human conditioned media with FGF4, HGF, and now the addition of dexamethasone and oncostatin M. By day 20, the cells had generated cytoplasmic vacuoles displaying morphogenic phenotype and had differentiated human hepatocytes. Now that concludes the technical part of my talk. At this point, I'd like to bring your attention to these hematopoietic cytokine bundles that we offer at GenScript that I discussed in my hematopoietic stem cell differentiation. Now, to show our appreciation of you attending this webinar, we're running an exclusive promo code, WebSC14, that will give you 15% off of these bundles or individual cytokines within these bundles from June 13th through July 31st. So I encourage you to take advantage. Also, I want to highlight GenScript cytokines. GenScript, we have a wide variety of recombinant proteins, and our purity is over 95%. We have low endotoxin levels with many below 0.2 EU per microgram. We have superior lot-to-lot consistency. Our specific activity is verified by a third party. And our prices are competitive. And we can do special requirements for, that fit the needs of our customers. So I encourage you to go to our website and look at our cytokine portfolio. And if you have any questions, please don't hesitate to contact me by email. So I want to thank you for your participation in this webinar and highlight some new webinars we have coming up. Also, due to popular demand, we are rerunning some of our previous webinars. So I encourage you to check our webinar website and see when those are being run. Now, exactly following this webinar, a brief survey will appear. I appreciate if you could fill out that survey. I always look forward to your feedback. And I wish you the best of luck in your research, and I have time for a few questions. So please, stick around. I will answer those. So we're just getting them up here. Okay. One question is there any advantage to using L929 conditioned media over MCSF specific for macrophage differentiation? So that's a good question. As I talked about, there's two methods to differentiate macrophages, using the L929 conditioned media or just using MCSF in your media. When I was culturing it, I was using L29 conditioned media, and I didn't see any differences from people that were just using MCSF in their culture media. The one thing to keep in mind, though, is when you're using the recombinant cytokine directly, you have more control over consistency. So basically, I mean, if you're using 50 nanograms per mil, you're always using the same amount. However, when you're using conditioned media, there is a slight risk for variation. However, with L29, because you're only looking at MCSF and you're not looking at other cytokines or growth factors, that variation is very limited as opposed to the variation you might find in FBS. But that's the one difference is, when you're using the recombinant cytokine, you can really control how much of it you use from each experiment. This is a question here. Are there any protocols available for mouse embryonic stem cell differentiation to hepatocytes? That's something I have to look into. I'm sure there are. We're going to have a whole stem cell protocol. I'm going to post them online. It's focusing on human embryonic stem cells, but I'm sure there are ones for most. They would definitely use different cytokines because you'd be using, if you're keeping them differentiated, you're not going to use the same protocol you would use for human. But I'll definitely look into that, and I plan to post these protocols online. Let's see, any other questions? 2 inhibitor system. What about a 2 inhibitor system? I'm not sure I completely understand that question. I do know some people culture embryonic stem cells using inhibitors, and that has been shown to be effective in preventing differentiation. So that's something else I will post on the website. I hope that answers your question. There are studies that show specific inhibitors will prevent differentiation in both human and mouse embryonic cells. So that's possible. So yeah, I'll post that. I'll take one more question. So will these slides be available? The answer is yes. So we're going to post the webinar and the slides online. They should be available next week. And you can download the video and download the slides for your use. So they should be up. Just check the website and see if they're there. So with that, I'm going to conclude today's webinar. Again, I appreciate everybody for attending, and I look forward to seeing you at future webinars. Thank you.